Dr. Syed, and he's going to be presenting on some genetic research that he and his team have been working on.
Yeah, excellent. Thanks so much for the introduction, and always glad to talk about hidradenitis, especially when it's to patients. That's always kind of the nicest venue to, try to get information out there and educate as many people as possible, and to get feedback too, because a lot of times when we, think about research, how to treat patients better, it comes from suggestions from patients and what are the things that you care about knowing. And so that's for me, one of the most important parts is having this dialogue, and for all the patients that volunteer to be part of the studies, that there's a way to get that information back out there, so that it's they know kind of what happened with that spit they put into a tube a long time ago. So I'm going to jump in. Let me go and get my screen shared to make sure I got things pulled up the right way. But hopefully I think you can see the screen up there, and I'm going to do some summarization of what our group has done with regards to HS and genetics research. And also, there's other groups that have done a lot of work on the genetics of HS too, trying to help understand, why it is that it seems like it's more common in certain families, and what role the genes play as opposed to what is happening in the outside world that's influencing the HS in some way. So I'll go ahead and jump into it here. For me, again, a lot of the questions that I think are sort of important to answer in research are things that came to mind because I see patients in clinic all the time. I come across the same types of questions about, trying to help understand why HS does what it does. And I always want to have those answers when I can. And so for me, early on, as I started seeing a lot of patients, I often got asked the question, is this something that I can pass on, right? Could I give this to my kids? Are they more likely to get it? Do I need to be watching out for them? And so, being able to do some research around the genetics of HS and how heritable is it? How much influence is there was an important question to try to get an answer to. Because I said, sure, I see a lot of patients with family members that have HS, but it's hard to say exactly how common that is and what the influence there is there. Sometimes when you see conditions clustered together, it happens in the same families in the same household because it is something that could pass between people. We know that HS doesn't do it that way, or we know that it could be something where something in the environment triggers a condition or people to get sick. And so the question was, is that clustering, how much of it is it related to genetics, the fact that people are related and share DNA, versus how much of it is something in the environment around them possibly, and trying to separate those two things out. And one of the first ways to try to do that was as I saw a lot of patients and we started asking about information about whether or not family members were affected. We found that, more often than not, so 50% of the time or more, actually, patients would say, yes, I have a first-degree family member. And it wasn't always, that my mom had HS. It was, anybody of that has HS, no. But then if I said, did anybody get boils when they were younger? The question was also yes, because again, that person might not have been diagnosed. And so asking those questions the right way became very clear that even if hidradenitis wasn't a diagnosis in the family, that there were other family members living with the same condition very clearly, that probably just hadn't gotten their diagnosis before. Maybe theirs wasn't as severe as the patient I was seeing. So more than half the time there was a first degree family member affected, so a parent or a sibling or offspring. And then even more often than that, we hear about, okay, yeah, mom or dad, no, but yeah, my aunt had it, my grandma also had it. So secondary relatives are also very common. So it became clear that there was probably this genetic susceptibility or genetic risk of some kind that was happening. And then we wanted to try to put some numbers to that. And so when we looked at a lot of patient pedigree, so we looked at 300 or 400 families and tried to sort of tease out, who had HS and who didn't. I was able to work with some geneticists on campus and do this calculation that it was about a 20-fold risk probably, where we know that in the general population, the rate of HS is somewhere between probably 0.5% and 1%. Some have said as low as 0.1%, so like 1 in 1,000. Others have said over 1%, and I think that's too high. So it's probably somewhere, about 1 in 200 people, 1 in 250 people, something like that. And so if that's the average risk, is if you said assumed high and you said it's 1% of people get HS in the general population, then if you've got a child or a sister or a parent, they'd be 20 times more likely to have HS than the general population would, which means maybe it's about a 20% chance that a child or a sibling would also have hidradenitis. That also supposes that everybody's hidradenitis transmits the same way, though. And it's much more complicated than that. There are some conditions where it's, you inherit one gene, I'm going to oversimplify it no matter what example I give, but we'd say that if you have, you can imagine there's a trait for like sickle cell disease, and it's more complicated than this actually. But if you get that sickle cell gene, then that means you're going to have some sickle cells in your blood. It's just controlled by that one gene alone. Whereas a lot of conditions can be controlled by many different genes that all lead to a similar outcome. And so with HS, it's not like there's one gene like that where every family's getting the same genes and the same risk. There's probably different risk in different families, actually. So, for example, there's what's called this gamma secretase complex, and there are 4 different genes that help create that gamma secretase complex. It's a protein in the body that helps to, help with lots of processes throughout the body. And if one of those 4 proteins is mutated for some people, and they have a copy that doesn't work the way that it should, it almost always has a very high risk of leading to hidradenitis. And there are certain families where they can track those mutations, and the people that have them, almost certainly get HS, and the people that don't are very, very unlikely to get HS. So, but if people go look for those mutations just across all hidradenitis patients, only about 2 or 3% of patients have those mutations, and their hidradenitis often looks very different. Often they've got a lot of lesions on places like their back and their chest. It's not really limited to places like underarms and groin, like we think of hidradenitis typically being. So, for those patients, it's a very sort of like, 50-50 chance. If the person, that's a kid gets that gene, it's very, very likely. But half the time they're not going to get that gene. But for most patients, it's probably more complicated. So sometimes you see HS come up out of the blue, or you just see maybe there's, of 50 family members, just 1 or 2 people who got it. So they have a low risk gene of some kind potentially. So, part of how I, try to answer that question for people is, on average, it's about 20% chance, a child of yours could develop hidradenitis if we just took it like that. If you have a very high rate in your family where everybody's getting it, then you're potentially a little higher than that on average. But if it's just you or just, very few other people, then that risk is probably very low, actually. And so not that much different than what it would be for most other people. So there's not like a one size fits all answer, but it's definitely, I think, more informative in that way. We can kind of think about it and sort of know what that average risk is at least. There's also similarly been a lot of twin studies. So some countries have twin registries where they basically have contact information for twins who are identical twins and non-identical twins. And they can ask questionnaires to try to help understand genetic diseases that way. And so kind of seeing what sort of risk factors are there, how much did the identical genes affect risk when they both would have HS. And I've got, a couple, more than a set of identical twins that both have hidradenitis. And so, I've seen that relationship in real life, but there is clearly a much higher concordance among twins and identical twins than there is against siblings who don't share the exact same DNA. So that also backs up that there is something genetic that's sort of triggering hidradenitis for a lot of people. And it also backs up the idea that, again, this is not like an infection or a contagious thing. It's not that people are passing it within their households. It's that there really are genes that are very important here. Just like if you look at psoriasis or rheumatoid arthritis or things like that. One of the sort of interesting things too about the gamma secretase genes, like there's actually some medications that block these proteins for treating certain kinds of cancers. And if you block those proteins, you actually can create hidradenitis, like people with on those medicines can get hidradenitis-like lesions. So it's a very sort of unique scenario. But these are some, responsible in a very small percent of patients. And so, one of the main projects that we worked on is what's called a genome-wide association study, or we abbreviate as GWAS or GWAS for short. And what that basically does is it tries to find in large populations, what changes in the genes are associated with a condition occurring more often. And so this is just an example here. You have these what are called like giant arrays where there's like, tens of thousands or hundreds of thousands of different individual genetic changes kind of mapped out across these sort of little tiny plates microscopically. So this is very much magnified to see these little tiny lights on the plate. And what you do is you test, patients with a condition versus those without for all these hundreds of thousands of different possible changes that could be occurring. This isn't even all the genes. I mean, there's things that even these huge arrays don't test for completely. And you look to see, are there certain changes that happen to be more common in the patients with a condition than without? So it's kind of highlighting this dot here. This dot here shows that this gene it's red, so it's a missing, there's a change here, again, that sort of shows up more commonly in the cases here than it does in the control group without the condition. And so if you do this on a huge scale, you find some things by chance maybe, but a lot of these things, if you find the right people to compare against, you see certain things that really start to stand out. And when you plot it out and try to record what it shows, this is this here is a gene that's much more common in the patients with the condition than without. So it starts to, this is how they measure whether it's important or not, or just like a tiny trend or like a very glaring difference that seems to stand out. So the higher up the dot gets, the more it stands out as there being a seeming difference between the group. And so again, we set out to do a study like this, done for lots of other conditions over time, found a lot of genes that are responsible for different conditions. We've had, over 1,400 people who have come into our clinic with HS that have agreed to be part of these kinds of studies. So it takes a huge amount of effort. And I'm always, super thankful that patients with HS are so willing to be part of helping to understand their disease better. For the most part, they spit into collection tubes. Occasionally, we can use things like a blood draw to also get DNA. And so we did an initial round of analysis on the first 720 subjects, and ends up being diverse, kind of like we see generally for HS patients. About half of the people were Black, 80% were female, and most had pretty active disease. So we say like 48% stage 2, 37% stage 3. So most people had more than like very early disease. And only about half of these patients had a first-degree relative that were affected. So we knew that there was a high family risk within these populations. And then you need a lot of people. The bigger the numbers are, the more you can start to find associations. And so we didn't just take our own patients. We also took patients from these other what are called biobanks. So there's other biobanks out there where they just take, every patient that comes into a system, they have them spit into a tube after giving a consent and agreeing to be part of something like that. So in Finland, they have this FinGen, which is a huge repository. And then they sort of link data. So we could find out that in this big FinGen repository, there were 673 people with HS who had donated saliva over time or donated DNA over time. Same for this one in the UK with 247 cases, another in Tennessee called BioView. And so by adding together all these cases, we had more and more power to try to find differences. And we used a different group from what's called AdHealth, a different repository, to find people without HS that had similar things like, age, race, sex, to make sure that we were kind of matching those things between the two groups the right way and finding true differences, not just related to some other background difference. And so this is what our initial GWAS looked like, was that you see that some of these dots start to rise above what are called the significance line. So these are, genes that are sort of differences in the genes that are found much more commonly in patients with HS than not. And so this all clustered here at the same gene. These are changes, there's multiple variations that can happen to the same gene. And so all these variations were in that same gene on chromosome 17. So we have 21 chromosomes. And this here is, again, 23 chromosomes. There's a couple up here. There's not a 23 on here, but this was at chromosome 17. This one here that's getting very close in the initial analysis was on chromosome 13. And so we'll talk about these two genes. There's a couple other that start to get close to being important. You can see these others that are kind of floating up here a little bit randomly that didn't quite meet it. And by the time we combined in a lot of other databases, like the other ones I talked about, because this was just our own patient, or these were the first couple of them, if you combine them all together, these became even more significant. And so these are some of the first genes that are thought to be related to HS beyond those gamma secretase genes. Understanding the genetics, I'm going to try not to lose people, because I've had to learn a lot about genetics, or at least try to make sure I kept up with all the geneticists that are part of this team, because it's a big group of people that helps do stuff like this. But there's the part of the genes that make the actual proteins, like the building blocks that help, make all the cells and things like that. So parts of the DNA have the genes themselves, and parts of the DNA next to it sort of have the instructions for when those genes do things like turn on and off. So it's the on-off switch for each of those genes. And so the changes we found are more in the regions that are sort of like the on-off switches, or change how the gene might be processed in some way. So it's not that the protein is necessarily messed up as much as how it switches off and on is messed up, probably. And what those differences, or those changes were very near to, were these genes called SOX9 and KLF5. These are acronyms that comes out to much longer names that I won't try to say here. But what's really interesting about these genes is that they seem like they're highly relevant. Like sometimes when people do studies like this, they find some gene and they have no idea what that gene actually does, and it doesn't seem relevant at all. But it was like if we found a gene that was important in neurons in the brain or something like that, and we say, we don't know what that means, because it's clearly it doesn't make sense that it would affect something in the brain and not the skin. But both of these genes are very important in the skin. In particular, the epidermis, that's kind of that top layer of the skin, and then the hair follicles. and even more particularly in the stem cells that regulate those things, that help a hair follicle know how to be a hair follicle and stay a hair follicle. And in the epidermis, that sort of that programs, how the skin does things like heal and maintain itself over time. And so, SOX9 is very important down here in the hair follicle where the hair grows from, and all these cells that help to maintain the hair follicle. And KLF5 is really important in the epidermis, so the very top layer of the skin. And the stem cells help sort of regrow parts of the follicle, or when healing needs to happen, if there's been a wound, those stem cells turn on to help with the healing process. And so normally what you have is you have SOX9 down here in the hair follicle telling that hair follicle how to be a hair follicle. And you have, in the epidermis, KLF5 that's helping to maintain the epidermis, keep its integrity. And when you have a wound in the skin, it's sort of normally, so if there was a trauma of some kind, normally SOX9 is only in the hair follicles, KLF5 is only in the skin, and KLF5 actually stops SOX9 from going out and changing things other places. So they sort of know how to turn each other off and regulate each other. But if that regulation is messed up, normally in wound healing, you have to heal a wound. And so both stem cell populations turn on, they're trying to fix everything and come in and sort of help that healing happen. But if KLF5 or SOX9 doesn't work the right way, normally what happens is at the end of wound healing, KLF5 turns SOX9 back off. So it says it's time to be skin again, turn this hair follicle stuff off. But when that doesn't happen for some reason, you can create chronic wounds. So people who develop things like chronic leg wounds will see KLF5 and SOX9 kind of turned on when they shouldn't be together. Normally, they should be very separate. The other time you see them turn on is in things like squamous cell skin cancers, where there's too much skin growth happening. The cancer cells are telling themselves to replicate quickly, so both things get turned on in that situation. And so it's really important they're able to do that to help heal a wound, but it's got to be able to turn off and get back to normal for the wound to heal itself the right way. So, what this tells me is that, there is something probably dysregulated with the body's ability to heal itself, so that once, something happens that causes trauma to the skin, so if there's some inflammation around, the body can't turn that off the way that it normally would. And it can't create sort of a normal skin lining. Again, instead of a skin lining, it does things like create these tunnels like we see in hidradenitis, where it's not the same as a hair follicle and it's not the same as a flat skin surface. You get these weird pockets and cysts that start to develop instead. So the structure of the skin changes with all the scar-like changes we see with HS. And the inflammation becomes chronic because that part doesn't turn off either. And then the other thing it probably does is it affects how the hair follicles maintain themselves. So if the hair follicles don't sort of maintain themselves the way they should, you can imagine that maybe those follicles are sort of more fragile. And so they have more of a tendency to do things like rupture, or maybe they don't contain things like, if there's things like the normal bacteria on the skin that live around a hair follicle, maybe they're not protecting the rest of the body from what's around the top of the hair follicle the same as they usually would. And so the body says, starts to feel threatened, or the follicles rupture. So the body overreacts over and over again. So there's some disruption of the maintenance of the hair follicles. And I think it would make sense that, the friction and trauma, just like we know that like clothes rubbing in certain areas will like trigger an HS lesion sometimes, that mechanical friction maybe is rupturing follicles even because they're more fragile for some reason. That leads to all that chronic inflammation. So to me, it was like, man, this feels more than coincidental that these genes, not only happen to be in the skin, but they happen to be really important in hair follicles and in wound healing and in maintaining, sort of the integrity of the skin. So this was, even though people had never really thought about these genes before for HS, it's like they fit in kind of perfectly as far as explaining why somebody might be more predisposed to getting HS if they've got issues with how this is regulated. And there have been some experiments in the past done as people were just trying to understand these genes, not recognizing they had anything to do with hidradenitis. And experiments there to try to understand them show that if you take SOX9, if you keep these genes from functioning at all before a mouse is even born, they really don't survive. They have too many different problems. But if you turn off SOX, because it's important for other things beyond just the skin too in very early development. But if you turn off SOX9 and mess up how it works once the mouse is a little bit grown and just in the skin, what happens is those mice start to have hair follicles that become fragile and that don't heal. So they start to lose their hair and not reform and lose the follicles over time. And they get a scar-like reaction and a lot of cysts. So like sort of where the follicles have dilated out and become cyst-like structures, which we can sometimes see in patients with HS. With KLF5, if you see things like too high, like KLF5 being too high, those mice get chronic skin wounds. They also lose hair follicles over time, probably because KLF5 is shutting off SOX9 and how it helps to maintain the hair follicles. And it's another really interesting thing, but you also see inflammation in the gut, kind of like Crohn's disease or inflammatory bowel disease. And we know that patients with HS are much more likely to get inflammatory bowel disease, actually. And what's also interesting about that is that you see that the gut and the bacteria change in those mice, like different types of bacteria tend to live. It's not infection, but there's a change in the gut bacteria. And if you give those people antibiotics or medicines like IL-17 inhibitors that block inflammation, just like some of the medicines we use in real life now, you make their condition better, actually. So it very much mimics what we see with HS, where there's this change in the skin bacteria. Being on antibiotics seems to help suppress the inflammation some. Maybe medicines that block things like IL-17 are helpful when this change occurs. So there's a lot of coincidences here that again sort of starts to sort of link things together and make sense. All right, so that's again maybe getting a little bit more complicated than it needs to be, but it feels very interesting to see how these genes potentially are affecting why HS happens. And next important steps are that we're just sort of scratching the surface. Anytime that we start learning answers to some of the questions that we're asking, it feels like it can make things even more complicated, like that there's a million more questions to ask. So we found 2 genes that seem like they're important probably. There are probably a lot more genes that if we just looked at more patients, we'd have found even more. And so there's been a couple of very recent studies that have been presented at meetings that aren't published yet that looked at more patients, so a few thousand patients, and found that actually there's probably a couple more genes that you can start to detect once you have that many people. So one of our next big goals is we're putting together proposals for a study to look at like 12,000 patients, going up higher on our own patients, about 2,000, and then sort of working together with other databases to combine even more subjects together. And so that's kind of in process now. And then so larger studies are going to be very important. And then it's also going to be maybe important to look at certain things. Like we know that women are about 75 to 80% of people with HS are women, and about 50% in my own population are Black patients. We know that Black people have about a 3-fold risk of developing HS compared to people of other races. And so of other sort of their ancestry, I should say. And so so there maybe are some differences there that predispose certain groups more than others that really don't look terribly different in these particular genes. So we can hopefully start to sort of figure out, are there, again, multiple genes that are important, and are there reasons why we see these changes in the population happen the way they do? So hopefully we get to answer more of those things. And we've got to prove, try to understand like what SOX9 and KLF5 are doing, these 2 genes we just talked about. It's not just that they're like completely off or completely on, probably. It's that they're regulated in a different way. And they might be working just fine in one type of tissue and sort of just messed up in the skin because of how it maybe is just dysregulated in some way. So there's some experiments planned to help figure out, when these changes happen, how does it affect how those genes are turned off and on, and to really understand that better. And ideally, again, that always raises new questions about, maybe if we can understand it better, can we figure out better ways to target treatments towards it, which is the ultimate goal. It's very nice to figure these things out and learn about it, but ultimately we can hopefully, get some better answers to some of these questions and really, maybe figure out better ways to treat when we understand it better. Now I try to give a shout out to the team that helped do this. Again, I am presenting it to you here, but Dr. Yun Lee and Dr. Karen Mulkey, they're the primary investigators on this. Dr. Mulkey is like a very well-known geneticist who does a lot of genetic research, and I was able to pull her in and talk to her about HS and get her to really take it on. And Dr. Yun Lee is super well-known when it comes to how to do all these analyses. Right, it's very complicated, all the computations that have to be done to figure out and do these things the right way. And these were some of the people that work in our labs along with us that have helped do a lot of the work. And then if you've happened to participate in our studies in CS and clinic, you maybe have met one of my research fellows over the last 6 years. They've each spent a year with me and helped collect all the saliva and organize all the samples, get them over to the labs, work with patients closely. And so I'm thankful to them, because none of this stuff can get done without a lot of people putting in a lot of effort. And then again, like I mentioned before, how thankful I am to all the HS patients for agreeing to be part of something like this. It's a totally voluntary thing. And I think just people who want to understand their disease better and help other people understand it better, so they don't have to suffer the same way. And then a lot of this funding for this project came from some internal UNC funding, but the majority of it came from the National Institute of Arthritis, Musculoskeletal, and Skin Diseases, which is part of the NIH. So this was done as part of the first NIH grant that was ever given to study hidradenitis in particular. So it was exciting to get to be a part of that big change. The NIH had not given any grants to HS research before 2019, which was a real kind of something that I think once it came to their attention made them feel really bad. But there were also not many people doing research who were putting those grants in there. So they really made a concerted effort to attract proposals and help spur research in HS, and it's just starting to pay off now. And so that's all I got, but I'm happy to try to take some questions.